So today we are approaching the last part of chapter 2 from the Cellular and Molecular Immunology book from Abbas, and we are going to discuss today about the lymphocytes. The lymphocytes are regarded to be one of the most important cells of the immune system, and it's extremely important to understand what are the main characteristics of the lymphocytes that differ these cells from the other cells of our body. So one thing that is very important to understand about the lymphocytes that I would like to highlight here is that these are the only somatic cells that we have in our body that can make or perform recombination in their genes. What? In order to understand this conception, we need to look back to the definition of the terms. So first, somatic is everything that we have in our body that's not the germinative line. So the germinative lines are the reproductive cells. Our eggs or the spermatozoids, they recombine naturally. They recombine the genes to induce variation in the genetic code, so it might provide survival of the species. This is fine. All the other cells of our body, they don't have recombination in the DNA. They don't have any differences in terms of the genetic code. The DNA is just the same in all of them. But what is the DNA? Imagine that one cell is like a kitchen. And what's the purpose of the kitchen? So in a kitchen, in order to produce and make food, you need to follow a book of recipes. So the DNA is the book, and the genes inside of the book are the recipes. So different genes are like different recipes to produce different types of food. Okay? So the purpose of a cell is to produce not food, but proteins or other complex molecules. But in order to produce those molecules, those proteins, the cell or the cells, they need to read a book, and this book contains different types of recipes. So all the cells of our body, they have the same book, the same DNA inside of the nucleus, okay? And these cells, these books, they also have the same genes, the same recipes. So the book is nothing but a collection of recipes that the cells will read, and the cells will make different types of foods or proteins in response to different environmental circumstances. That's why some cells are like physically different, because they are in different parts of our body. They express different recipes, but the book is the same. The DNA is the same. It doesn't change. It's exactly the same. The DNA of a cell in our head is the same of the cell in our foot. It doesn't change. It's just the same. Okay? So inside of our body, there is one type of somatic cells that has a DNA with the proclivity to change or recombine the genes. It's the only type of cells that does that. And this type of cell is called lymphocyte. So there is one particular part of the book and a group of genes that they can recombine and make new recipes to produce new proteins that will react and fight pathogens that we never saw before. So we are talking here about virus eventually that doesn't even exist. So these cells are the only ones capable to do that. And it's unbelievable. The capacity that the lymphocytes have to produce proteins that can recognize different types of pathogens is unbelievably large. We are able to recognize and produce proteins against hundreds of millions or thousands of millions of pathogens. And what are the classes or types of lymphocytes that we know so far? The B cells and the T cells. So regarding this table here, the most important cells that you need to understand, they are the CD4 helper cells. They receive this name helper because they will help the immune response, the type of immune response. So depending on the pathogen, if it's an intracellular infection or if it's cancer, these T helper CD4 cells, they will constitute a T helper type 1 immune response or a T helper type 2. Recapitulating, a Th1 or a Th2 or even a Th17 and so on. So the type of immune response, it's orchestrated by these cells here, the helper T cells, the Th or T helper cells, and they mostly express a formula or a recipe in the surface of their cell, which is the CD4 molecule. I will show this molecule later. So they are the most abundant ones in the blood, in the lymphoid organs where the immune response happens. So look at the numbers, they are extremely important cells. Other ones are the CD8 cells. So the CD4 are the ones that orchestrate the immune response. They secrete proteins, formulas, recipes. They tell and connect different proteins and cells together. They attract other factors. They constitute all the inflammatory field. They organize the inflammation. And the CD8 cells, they don't organize anything. They are mostly effector cells. They just arrive there to fight. Hey, I'm here to destroy whatever needs to be destroyed. So they express a different signature, a different recipe in the surface of their cap, in the surface of this cell. So they look like CD8 cells. This is how they look like. They are the ones that will arrive in the place of inflammation, that will kill the cells that are infected with virus, bacteria, or even tumor cells full of mutations. They will just destroy the reservoirs of infection. They are cytotoxic cells. We also have an important type of T cell here, which is the regulatory T cells, who is nothing but also a type of T helper cells. So the T regulatory cells is also a T helper CD4. But instead of behaving the Th1 type or Th2 type or Th17, whatever, it behaves in a regulatory process. These cells are the ones who deactivate the immune response. So the T helper are the ones that activate the immune response to different types of immune response. And the T regulatory cells, they are the ones who shut down the immune response. Because, well, you must agree that at some degree the pathogen will be eliminated, or the cancer cell is just eliminated. We can't keep the inflammation forever. So we need to recruit these dudes to restrain and contain the inflammatory process. Okay? So then we have like some types of rare cells, the natural killer T cells. They are important to kill tumors and also virus-infected cells. We're going to discuss about them. The gamma delta T lymphocytes, they are both helper and cytotoxic in the same way that mucosa-associated invariant T cells. And in the B lymphocytes group, the most important ones are the follicular B cells, because they are the ones that are located in the blood and also in the lymphoid organs. They are highly numbered in the spleen and also in the lymph nodes. And then they drop in the blood to produce and secrete antibodies to neutralize pathogens that are in the blood. So that constitutes what we call humoral immunity. So the lymphocytes that are produced in the blood, in the bone marrow and drop in the blood in the lymph, they will also visit the lymphoid organs constantly in different parts of our body to try to find pathogens that they could react with. So if there is one pathogen in one particular lymph node here, but the only possible clone of T cell capable to react with this pathogen is in another part of our body, we need to wait. This lymphocyte will start migrating and visiting different lymph nodes until the moment that this clone will arrive here and with what, hey, I found a pathogen that I can fight against. This process is called recirculation of naive T lymphocytes between the blood and secondary lymphoid organs. So I hope that you enjoyed this lecture, and in the next one we are going to dissect the main functions and also activation and regulation process of the lymphocytes. Don't forget to subscribe in the channel and also share with your friends, and I hope to see you in the next video. Bye bye.